Hello, everyone. My name is Stephen Humphrey, and I'm a biology and environmental science student at Worcester State University. I'm one of three other students studying ecology under our faculty advisor, Dr. Diana Sharp, and the presentation title for today is The Analysis of the Macroinvertebrate Assemblages Throughout the Urbanization Gradient of the Tatnuck Brook of Central Massachusetts. And I know that's a pretty dense title, and there's a lot to unpack, so let's do just that. So urban streams consistently show ecological impairment, and I bet that's no surprise to many of you. But something very useful to note is that freshwater ecosystems are exceptionally susceptible to any effects in their watershed. They're said to be one of the most threatened ecosystems in the entire world, and as such, it's incredibly useful to know as much as we can about them. Urban streams have shown consistent enough responses to urbanization that experts have coined the term the urban stream syndrome to describe its effects. And these effects aren't just limited to ecology, they also describe water quality changes, hydrological changes, and hydrogeological changes. So there's a lot to it. And just as many ecosystem services are provided by these urban water bodies, again, it's very important that we know a lot about them. And that's where macroinvertebrates come in. Macroinvertebrates is a blanket term used to describe animals which lack a backbone. For our purposes, we sampled benthic macroinvertebrates. These are mostly insects in their larval stage, which reside in the bottommost substrate of aquatic systems, or the benthic layer. Macroinvertebrates provide a plethora of functional niches for their ecosystems, and as such, they are an exceptional tool to provide insight into the ecological state of the aquatic system. On top of this, a lot of macroinvertebrates already have established pollution tolerance values from previous studies, and again, as such, they're incredible indicator variables. Macroinvertebrates have been shown to respond to even low levels of urbanization, so it's a perfect use for the study. For our purposes, macroinvertebrates are perfect because what's special about Tatnuck Brook is that there's an urbanization gradient through it. And the three sites that we sampled at, Cook Stream, Pat Stream, and Cove Stream, they increase in urbanization respectively. There's already been evidence showing of this urbanization gradient with water quality variables, specifically salinity, and here's a GPS photo on the right of an aerial view of the Tatnuck Brook. Here you get another view of the Tatnuck Brook, this time in the form of spatial analysis showing the land use of the watershed. Here you can see qualitatively the differences in developed land use at the three streams, with Cooks being on the top, Hatch being in the middle, and Coes at the bottom. And here you have a bar graph of the land use data a little more isolated to the specific subbasins of each stream that we sampled in. At Cooks, the one at the top, you see the least amount of urbanization, and Coes, the one at the bottom of the land use map, you see the most amount of urbanization. Sampling in an urbanization gradient is incredibly useful because it takes away any spatial variability that you'd have and allows you to isolate the effects of urbanization much better. Now for the actual sampling, we elected to do quantitative sampling using a Cerber sampler with a 500 micron mesh. I am aware of some studies which say that you can get semi-quantitative comparable results with a KickNet, but as this is our first bout in macroinvertebrate research, we wanted to omit any error which could come from sampling, and as such, we just did quantitative sampling with a fixed area Cerber sampler. After collecting the samples, they were stored in 95% ethanol. This was done to avoid any dilution, which may not preserve them to the extent that we wanted them to. And this was also done in tandem with testing for various water quality parameters, including pH, turbidity, salinity, dissolved oxygen, temperature, and various nutrient sampling as well. After taking the samples back to the lab, we assessed if the samples were sparse enough to analyze them in totality, or if we had to do subsampling. And when we subsampled, we elected to do it in a combination of two different methods, which included random grid subsampling and fixed count subsampling. After being sorted using a shroud type or a Bulgarov sorting tray, the specimens were identified to the lowest taxonomic level possible, with an emphasis specifically on family. And for the identification, we used dichotomous keys. Dichotomous keys are tools which allow you to identify something based off the presence or absence of either behavioral traits or morphological traits. But as our samples were sitting in 95% ethanol for a couple of months, we found it kind of hard to utilize behavioral traits, and as such, we relied mainly on morphological features. For an example, all these photos I have in this slide are of the same specimen, but the photo to the right, which is the specimen's head under a 100 times light microscope, um, was the only uh, was the only time where the features became discernible enough to identify it to family. So identification is a very involved process, but with that, I learned a lot and it was very useful. The identification was also aided by lectures. This was a picture of our first lecture. Thank you very much to Dr. Robert Smith from the Mass DEP. His insight was incredibly helpful, and I can't thank him enough. Just as I said before, macroinvertebrates are a great response variable to changes in water quality. Specific indices use attributes about certain taxa to assess the state to an even more scrutinous extent. For this project, we used EPT taxa richness, and EPT calculates a relative abundance of a certain group of 3 orders of macroinvertebrates compared to the total taxa found. This is one of few indices which has been shown to show consistent responses to urbanization, and as such, it's a great variable to assess the urban stream syndrome in the Tatnuck Brook. Now, in the name of the indice, what does EPT stand for? Well, EPT is an acronym for 3 different orders of macroinvertebrates. They are Ephemeroptera, Plecoptera, Trichoptera, respectively. Now, these 3 orders are special because throughout their taxonomic resolution, they all show relatively low tolerance to pollution. As such, using them in the indice, putting the taxa of EPT over the total amount of taxa is a great response variable to urbanization or any changes in the watershed. Here's everything summed up. After analyzing EPT taxa richness using R for each sample rather than each site, a linear regression model was run using the indice on the y-axis and the percentage of developed land or urbanization on the x-axis. The result is a model with an impressive statistical significance and a p-value far lower than 0.0005. Basically, summed up, this model shows that as land use is more developed in a river subbasin, the EPT taxa richness goes down significantly, and as such, the ecological integrity is impaired. Now, what's the significance of these results? What do they mean? Well, this research further establishes the evidence between land use and ecological impairment, especially in urban streams. It also further supports evidence of the urban stream syndrome and shows that there is evidence of the, um, the Tatnuck Brook suffering from the urban stream syndrome. Lastly, it further reinforces the evidence that there is an urbanization gradient in the Tatnuck Brook, and as such, it allows for further analysis of urbanization's effects on streams. Further studies. Now that we have all the data, we can do a lot with it. For instance, a multivariate regression may be done with the water quality parameters that we analyze in tandem with the macroinvertebrate sampling. This would allow us to deduce what specific symptoms of the urban stream syndrome is causing this ecological integrity to be lost. On top of that, we may do an analysis on land use change over time. This would be done to see how immediate the effects of urbanization are on these aquatic systems. Lastly, look into the higher trophic levels, such as fish, may be useful, as we already have insight into the response from macroinvertebrates, which are many of these fish's food source. It would be very useful to see the response in higher trophic levels to get more information on the totality of the urban stream syndrome. And here are the references we use for the project. And lastly, to just say thanks for a lot of the people who allowed this project to happen. I'm very, very lucky to be a part of this project, and I can't say how great of an experience it was. This was a huge, huge collaboration between a lot of different colleges, a lot of different organizations, and again, I can't thank anybody enough who was a part of this project. I do want to give special thanks, however, to my faculty advisor, Dr. Diana Sharp, both instilling a passion for research in me and making the entire process a lot less intimidating. And of course, thank you to Mass URC for allowing me to present, and thank you for watching.